Welcome to the deep dive. Today, we're plunging into an everyday puzzle, really, something that probably resonates if you've ever tried to grow, well, anything.
That mystery of why things sometimes just don't grow evenly.
Exactly. We're digging into some observations from Notes on Fishkeeping, Part 133 specifically, a piece called The Mystery of Uneven Growth of Common Salvinia.
Ah, Salvinia. Yeah.
It's this personal account from someone named Ang who's meticulously trying to figure out why their aquatic plants are doing so differently in spots that seem, well, pretty similar.
And what's great is how Ang's notes sort of mirror the scientific process.
Yeah.
Observation, hypothesis.
Yeah.
It's all there, even on a personal scale.
Right. So our mission today is to really unpack Ang's insights, look at the factors they're considering, and see what it means for understanding plant growth, maybe even in your own garden or fish tank.
Yeah, let's get into it. It's fascinating how these small observations can lead to bigger questions about environmental interactions.
Okay, so Ang kicks things off by talking about their routine. They regularly prune their common salvinia, moving them between different outdoor ponds, they call them.
So active management. They're not just letting things run wild.
Oh, not at all. And you'd think, okay, if you're moving them around, pruning them, things should be pretty consistent.
You would think so, yeah. But that's the crux of it.
It's precisely not what Ang is seeing. The core mystery, as Ang puts it, is these different growth cycles and densities in each of the ponds.
And not just slightly different, significantly different, you said.
Yeah, significant. Like picture one pond just absolutely choked with green, healthy salvinia, and then maybe the next one overlooks sparse, struggling.
Even though to the eye, they might look like they get the same sun, same water.
Exactly. That stark difference is what got Ang investigating. What strikes you about that starting point?
Well, it's Ang's persistence, really. They're managing the plants, intervening, but still observing closely and asking why. Why the difference despite my efforts? That's key.
It pushes past just accepting it.
Right? It immediately makes you think, okay, what are the unseen factors? Ang's first thought, sunlight exposure plays a major role.
The obvious starting point for plants.
Totally. Photosynthesis runs on light, but it's often more subtle than just sun versus shade, isn't it? We're talking microclimates.
Ah, yeah, like tiny differences in light duration or intensity.
Exactly. A nearby wall, a tree branch casting a shadow for just an hour longer, maybe even the reflection off the water, changing things slightly. What looks similar to us might be a whole different world of light for the plant.
That's a really good point. Our scale isn't the plant's scale.
Precisely.
And Ang doesn't just stop at sunlight, which is smart. They broaden the scope.
They mention the potential impact of different water parameters and fish populations as contributing factors.
So bringing in the water chemistry and biology of the system.
Yeah, it's that ecosystem thinking. When we say water parameters, what does that really cover?
Good question. I mean, it could be a whole range of things, right?
It really could. We're talking pH, how acidic or alkaline the water is. Plants have their preferences,
Right. And nutrient levels, too, like nitrates and phosphates.
Definitely. Crucial food for plants.
Yeah.
Too little, they starve. Too much.
Yeah.
Well, you might get an algae bloom instead.
Okay. What else falls under parameters? Oxygen? Temperature?
All of the above. Dissolved oxygen for root respiration, water temperature affecting metabolism. It's all interconnected. And, importantly, mostly invisible without testing.
So a lot of potential hidden variables just in the water itself. Then Ang adds fish populations. How do fish play into this?
Fish add another layer of complexity. I mean, on one hand, their waste is fertilizer, right? Nitrates, phosphates, good for plants.
Okay, so they help feed the plants.
They can, yeah.
Yeah.
Plus, they release CO2, which plants use. So a healthy fish population could actually boost salvinia growth.
But there's a flip side.
Oh, absolutely. Some fishy plants, herbivores, could be munching on the salvinia, keeping it down.
Oh, okay. So they could be the culprits in the sparse ponds.
Potentially. Or even just stirring things up. Bioturbation affects clarity, maybe nutrient release from the bottom. It's a balance.
Wow. Okay. So sunlight, water chemistry, fish.
Yeah.
Lots of possibilities. But Ang's notes seem to narrow it down, don't they? They focus on something specific.
They do. And this is where it gets really interesting because it involves Ang's own actions. They state, however, based on what I've read, salinity may be a significant factor.
Salinity. Salt levels. In freshwater ponds.
Well, potentially slightly brackish, or maybe treatments are adding salts. Ang connects this directly to their own management, saying they gave each pond different treatments based on my estimated dosages.
Ah, So Aang realized they might have accidentally created different salinity
It
levels.
seems that way. It's a moment of self-reflection, linking a specific parameter salinity back to their own actions, those estimated dosages. It's quite insightful.
Okay, so why is salinity such a big deal for a plant like Salvinia? How does that work?
It mostly comes down to osmosis,
Yeah,
how water moves across membranes.
right. Trying to balance concentrations.
Exactly. Plants need to pull water into their cells. If the water outside is saltier than inside the plant cells.
Water gets pulled out of the plant.
Precisely. Even if it's sitting in water, the plant can effectively dehydrate. It causes immense stress, makes it hard to function, hard to grow.
Wow. So adding slightly different estimated dosages of something could have inadvertently made some ponds too salty for the Salvinia to thrive.
That seems to be Ang's working theory, or at least a strong contender. It really highlights how our interventions, even small ones, can become really significant variables.
Yeah, you become part of the experiment without maybe realizing it at first.
Absolutely. It speaks to the need for, careful measurement if you really want to control variables.
So Eng's thinking has progressed. Observation, broad hypotheses, then zeroing in on salinity linked to their own actions. What's the plan now? How do they test this?
Well, interestingly, the next step Ang proposes isn't directly testing salinity. It's to start with introducing and increasing the fish populations to those ponds with less common Salvini population.
Huh. So they're going back to the fish hypothesis first. Why do you think?
It could be practicality. Maybe adjusting fish numbers feels more straightforward or immediately doable than trying to precisely measure and alter salinity across several ponds, which might need, testing kits, specific salts, or water changes.
That makes sense. Testing the biological factor first.
Yeah. They're basically asking, okay, if I add more fish to the struggling ponds, will the extra nutrients or CO2 kickstart the salvinia? It's a direct test of that fish population hypothesis.
It's a classic experimental approach, isn't it? Observe, hypothesize, then change one thing and see what happens.
Exactly. Isolate a variable. Even this backyard pond scenario, Ang's applying that principle. It's a great example of systematic thinking in action.
You really see the process unfolding in their notes.
And that systematic approach, that's what elevates it. Planning to add fish specifically to the less populated ponds allows for a comparison. You compare those ponds to the ones left alone or the ones already thriving.
Creates a sort of control group, informally.
Right. It helps tease out whether the fish are making the difference, rather than just guessing. It moves from, I wonder if, to, okay, let's actually test this.
So wrapping this up then, we've followed Aang's journey here, observing this really noticeable, uneven Salvinia growth, even though they were actively managing the plants.
Starting with broad ideas, sunlight, general water stuff, fish.
And then that key moment of insight, connecting potential salinity differences back to their own estimated dosages of treatments.
Yeah, that self-awareness is crucial.
And finally, the plan to actually test one variable, the fish, by adding more to the struggling ponds. It's a concrete experimental step.
And what this whole thing really drives home is just how interconnected everything is, even in a small pond setup. You nudge one thing, it affects others.
Definitely makes you appreciate the complexity.
It does, and it encourages us, to look beyond the obvious when something puzzling happens, to ask what else could be going on.
And to consider our own role, like Aang did with the dosages. How are we influencing the system we're looking at?
Exactly. It's a great prompt, doesn't it? Makes you wonder. What hidden variables are shaping things in your own world? Your garden, your work, anything. And, how could you maybe start to investigate them systematically, like Aang?